AP 120, Chapter 14, Topics: Cellular immune response and humoral immune response. So moving on to the third line of defense. This is our specific immune response. This can be cellular immune response or humoral immune response with immunity, meaning that you're getting a response mounted against a specific recognized foreign molecule, or antigens. So something on the surface of the pathogen is what we are responding to. Remember, our immune cells do not have eyes. They have to go around touching things to recognize them. So antigens. Antigens can be proteins, carbohydrates, glycoproteins, glycolipids. Again, they just have to be on the surface of the pathogen, the surface of the virus or bacteria or protozoa, and they have to be able to elicit immune response, which means that within our genes, we have this antigen cataloged so that we produce lymphocytes that will recognize that shape, that will recognize that antigen. So in our genes already. So before we're born, our immune response will take an inventory of self-antigens. What antigens are on the surface of our cells that are supposed to be there? Are structures that our white blood cells will touch and say, yes, this belongs here, this is supposed to be here. They take this inventory to make sure that we do not target our own antigens. So this is to prevent the immune response from targeting our own cells. That would be SPAD. And for example, a type of antigen that gets inventoried this way is, of course, the antigens on the surface of our red blood cells, such as antigen B. So if you have blood type A, sorry, that's an A. Antigen A. So if you have blood type A, you have antigen A on the surface of your red blood cells. So your immune response knows that's supposed to be there. That is a self-antigen. All right, so the lymphocytes are the cells, the leukocytes important for our specific immune response. Again, made in the red bone marrow. T lymphocytes mature in the thymus. They make up the bulk of the lymphocytes. And then the B lymphocytes and natural killer cells are produced from the red bone marrow, already mature. So B and T cells will go to their target lymphatic structures. This could be tissues in the walls of an organ. It can be lymph nodes, lymph nodules, tonsils, spleen, etc. And there they will screen for pathogens. So T cells, how do they work? So T cells and the cellular immune response. Well, strangely enough, it starts off with macrophages. So macrophages, neutrophils, the phagocytic cells that engulf pathogens, after they engulf and break down that pathogen, they will actually show on their surface antigens from that pathogen. So when they break up a bacteria, tiny pieces of that bacteria end up on their surface displaying these antigens. And they're always associated with the macrophage's major histocompatibility complex, which is just some other structures on the surface of the macrophage. Why do they do that? It's so that other immune cells realize that this isn't an enemy cell. This is one of their own cells displaying an enemy's, a pathogen's antigens. All right, eventually a T lymphocyte will bump into the macrophage. Again, touching is how the leukocytes get to know each other. And if that T helper cell recognizes that antigen, that becomes a significant interaction. It will then activate that helper T cell. So that helper T lymphocyte is designed for this specific antigen. If it comes in contact with a macrophage or neutrophil displaying that specific antigen, then the helper T cell is activated. If this was a different antigen, then nothing would happen. So if the antigen fits the T cell's antigen receptor, again, we get that perfect match, then the helper T cell becomes active. So again, here's the macrophage. It's displaying part of an antigen. Again, this is just a structure that the pathogen had on its surface that the macrophage is now displaying. Helper T cell shows up, And look at that, the helper T cell's receptor fits that antigen perfectly. Helper T cell gets activated because that helper T cell knows the pathogen and is ready to get the fight against that pathogen started. So, after the helper T cell is activated, it will secrete cytokines. Cytokines are chemicals produced and released into the tissues and bloodstream. One thing the cytokines do is stimulate the helper T cell that's active to divide. So now there are more of these helper T cells, all ready to fight, to help cause the fight against the pathogen. Also, cytokines will go out and increase the activity and the production of other leukocytes. So there will be more white blood cells moving through the body, surveying for pathogens. Cytokines also stimulate B lymphocytes, but only B lymphocytes that target that specific antigen. So again, this is a specific immune response. So, helper T cell gets activated, produces cytokines, activates other cells, including cytotoxic T cells, but only the cytotoxic T cells that matched with that antigen. So again, we're only targeting the cells that want to fight the specific pathogen that is now known to be present in the body. So the helper T cells activate and help the cytotoxic T cells to divide. Cytotoxic T cell then divides, producing lots more cytotoxic T cells, as well as some memory T cells. The cytotoxic T cells that are now active against that specific pathogen will go out and find body cells, our own cells, infected by that pathogen, and then destroy those cells. So the cytotoxic T cell is to destroy any of our own cells that are infected by this specific pathogen. So if the pathogen is influenza, then the helper T cell want to activate other cells that will fight influenza. So it'll only activate the cytotoxic T cells meant to fight influenza, and they will divide and produce lots of cytotoxic T cells that are destroying our own body cells infected with influenza. If it comes across a body cell infected with, say, Ebola, it'll ignore it, because the cytotoxic T cell only cares about cells infected with influenza. All right, the way to do it, the cytotoxic T cells release those proteins called perforins that then destroy the infected cell, similar to how natural killer cells function. And this requires cell-to-cell contact. So this is why this is a cellular immune response. Our own cells, the cytotoxic T cells, have to find the infected body cells. And again, the only way they can recognize them is by touching them and then destroying them. Memory T cells. Memory T cells are also produced. They are important because they will remember that we've been infected by this pathogen, so that if we get exposed to the pathogen in the future, we will cause a response much, much faster. So memory T cells make sure that we respond a lot faster to future infections by this specific pathogen, say influenza, and with the hopes that those future infections will be wiped out so fast that we don't even get sick. All right, then we have humoral immune response. Humoral immune response involves B cells. So we have the B cell with its own receptors looking for that antigen. So the B cell finds the antigen on the surface of a pathogen or on the surface of a macrophage, and that activates that B cell that is going to target that specific pathogen. The helper B cell then gets further stimulated and activated by cytokines produced by the helper T cell. So again, helper T cell stimulates the B cells activated by the exact same pathogen, the exact same antigen that the helper T cell was. The B cells then divide many, many times, and as they divide, they become either plasma cells or memory B cells. Mostly plasma cells, but also some memory B cells. So it's not 50-50, it's more like 90-10. And this then leads to the antibody-mediated immune response, also known as the humoral immune response, because the plasma cells will produce antibodies that target the original antigen. So the plasma cells are now producing antibodies that are targeting the structure on the surface of the pathogen. So again, if this is influenza virus, then the antibodies will bind to the surface of the influenza where the antigen is located on the influenza's surface. So antibodies move around in blood and other fluids and then eventually attack the pathogens. This is sort of an indirect attack. The plasma cells never have to come anywhere near the pathogen. And so that's why it's called humoral, so blood-related immune response. Again, here's the steps to antibody production. As you bring agents enter tissues, B cells encounter antigen that fits alone or usually with the help of helper T cells. B cells are fully activated, divide many times, becoming plasma cells and memory B cells. And then the plasma cells produce antibodies that target that antigen. That's going to bind to the pathogen to inactivate it and target it for destruction. Memory B cells act just like memory T cells. They remember that we've been infected so that in future infections, the response will be super fast, hopefully preventing us from getting ill. And that is it for this part.